बिस्मिल्लाहिर्रहमानिर्रहीम। तो आज हमारा जो क्लास है ये एक थ्योरेटिकल पार्ट है। इसमें हम थोड़ा सा डिस्क्रिप्शन भी देंगे कि लैब में या लेबोरेटरी में हम कैसे डिटरमाइन कर सकते हैं हाउ कैन वी आइडेंटिफाई द बायोलॉजिकल पॉलीमर्स लाइक कार्बोहाइड्रेट्स, ग्लूकोज, सक्रोज, फ्रैक्टोज, स्टार्च। तो ये सब बायोलॉजिकल पॉलीमर्स है। हाउ कैन वी डिटरमाइन दैट एक सैंपल में देयर इज प्रेजेंस ऑफ ए बायोलॉजिकल पॉलीमर। तो इसके लिए एक सिंपल सा टेस्ट है दैट्स कॉल्ड एज अ मॉलिस टेस्ट। सो वी डिटरमाइन दिस बाय मॉलिस टेस्ट। ठीक। सो मॉलिस टेस्ट में हम क्या करते हैं, वी टेक आवर सैंपल। उस सैंपल में लेट अस से वी हैव ग्लूकोज प्रेजेंट। सैंपल को वी आर adding to this sample the sulfuric acid concentrated. So add concentrated sulfuric acid to your sample. Then what you will see, and along with this we also add Molish reagent. Molish reagent. What's Molish reagent? It's alpha naphthol. So what is the structure of Molish reagent? This is the structure of alpha naphthol. This is called alpha position, or this is also called alpha position. This is called a beta position. So this is alpha naphthol. So when you do these things, you add these two things to this. What it does, you get, let us say you are doing these things in a test tube. So what you will observe in your test tube is that you will see the two layers here. So you will see two layers here. At the junction of the two layers here, you will see a purple ring. Nice practical purple ring. If the purple ring is present here at the junction of these two layers, that purple ring indicates that the sample contains your biological polymer, that is carbohydrate. If there is no purple ring, that means our sample doesn't contain the polymer carbohydrate, that's glucose or whatever. ठीक। Now इसमें होता क्या है, how can we proceed in the lab in the steps, what are the chemicals and glassware required, let us see. So this was the basics. Now so what we do is we require, number one, let us see the reagents and chemicals. Reagents and chemicals, or you can say reagents, chemicals, glassware. So glassware में क्या होता है, you require a test tube. Very simple. Take a test tube. You can also take pipette. Pipette जिसे आप ड्रॉप वाइज कोई चीज डाल देती हो ना, pipette, a pipette or a pipette. And then you require, it's a very simple practical. You require chemicals like sulfuric acid, concentrated sulfuric acid, concentrated acid जो मार्केट में होता है. Then you require Molish reagent. इसको मैंने कहा था, it's present in the market, Molish reagent. अच्छा, Molish reagent, it's in this practical we use 5% of alpha naphthol. That is very important. यानी जो मार्केट में अल्फा नेप्थॉल है उससे हम बनाएंगे 5% of alpha naphthol. किसमें बनाएंगे हम? इन इथेनॉल. ये बहुत इंपॉर्टेंट है. So that's your Molish reagent. Fine. So you require sulfuric acid, you require Molish reagent, and you require the sample जिसमें आपको डिटेक्ट करना हो. So we'll take in this case glucose, hexose sugar, 6-carbon sugar. So these are the reagents, chemicals, glasswares required. Now what we'll do? So we'll do this in the step number 1. So add 2 ml of your sample. Add 2 ml of your sample in a test tube. Step number 2. So add 2 ml of sample in a test tube. And the step number 2. To this add Molish reagent. molish reagent. ठीक, how much molish reagent? Take 2 ml of the molish reagent. So take these 2 ml and then add to this test tube, sample test tube. And then what you do, उसके बाद अब बहुत important, उसके बाद gently add concentrated sulfuric acid. How much? Concentrated sulfuric acid you can gently add few drops, few ml or 1 to 2 ml. But यहाँ पे याद रखना है कि for example this is our test tube. Sulfuric acid we have to add here through the walls, so that should slowly come down यहाँ पे. So with the help of test tube, so through the walls we have to add this. So gently add concentrated sulfuric acid along the sides of test tube. ठीक. तो what you will see, जैसे हमने ऐड किया, you will observe 2 layers forming, 2 layers, 2 layer बनेंगे यहाँ पे. यहाँ पे you will see that 2 layers are forming. This is layer number 1 and this is layer 2. ठीक, these are the 2 layers formed here. This is layer 1, this is layer 2. 2 layers, you will clearly see 2 layers. So अब आप क्या देख रहे हो उसके बाद, जैसे 2 layers आपने डिस्ट्रिक्ट देख ली. Observe any color changes. Observe any color changes. Color changes at the junction. At the junction. तो junction means इन 2 layers का जो meeting point है ये वाला. यहाँ पे आप नजरें यहाँ पे रख लीजिए. You'll see that some color change will there. If you see purple color in the form of a ring, purple color there appears, purple color appearance, it indicates presence of. So you will confirm that indicates presence of your carbohydrate or for example glucose. ठीक. अब क्वेश्चन आता है, how this purple color is formed, how actually, what is the reason that we're getting this purple color at the junction. So उसको understand करने के लिए we have to go to the chemical reactions involved during this process. I will not go into the mechanism of these chemical reactions, लेकिन what are the chemical reactions involved, let us see. अगर किसी को mechanism चाहिए, he can confirm me separately or he can write me at some mail and then we'll do it. So what happens, we are taking sugar, let us say our molecule was glucose. So we are taking our sample glucose. Glucose structure is like this. It's a 6-carbon compound hexose. Then you have CH2OH. Then this is CHO. Take OH. This is 1, 2, 3, 4, 5, 6 carbon open chain glucose structure. So what we are basically adding? We are adding sulfuric acid. Sulfuric acid is a concentrated sulfuric acid is a dehydrating agent. इससे क्या होता है कि water remove हो जाता है. So in this case, ये जो alcohol है, group है, and this aldehyde, they undergo reaction between this and this. उस reaction से there is loss of water. And we will get a ring compound. ठीक? So that's why it's called dehydration. So by adding sulfuric acid, we are basically doing the dehydration. Dehydration of sample, that is sugar molecule. Sugar. ठीक? So we'll get a type of a structure. I will not go into deep of it, जो मैंने पहले भी कहा in the mechanism, but up to the basic understanding, we'll get this type of a ring. This is called as a furfural ring. Furfural 1, 2, 3, 4 carbon ring and then 1 oxygen 5 member ring furfural. अब इसमें ये जो CH2OH एक point पे यहाँ पे we have this CH2OH which is here and then this on the other side we have this what we call as we have the ring is between this and this, so it's basically, since we have 4 carbons only in the ring, so it's between this and this. In दो molecule के बीच में we have dehydration, so that we have 1, 2, 3, 4 carbons, and then water molecule gets lost, so it's here, and here you have CH2OH on this point, and here you have an aldehyde group. So aldehyde group, that's this one, and CH2 is this one, so this is our compound formed. This is called as, if we do the numbering, it's 1, 2, 3, and 4. It's 4 hydroxymethyl furfural. This structure is called as a furfural, this. So it's called 4 hydroxymethyl furfural. अब so what we are adding molish reagent. तो मॉलिश रिएजेंट इसी रिंग के साथ रिएक्ट करता है, इस कंपाउंड के साथ रिएक्ट करता है आपका जो मॉलिश रिएजेंट है, दैट इज योर अल्फा नेप्थॉल. तो यहां में जस्ट बेसिक विल गो टू द बेसिक्स. क्यों हमारे पास विल गेट अ पर्पल कलर. सो अल्फा नेप्थॉल का स्ट्रक्चर होता है दैट इज this is our alpha naphthol. So alpha naphthol में इस we know that carbonyl group it can undergo the nucleophilic addition reaction. So इस कार्बन पे 2 molecules के react करते हैं from here. इस position पे mechanism में नहीं जा रहा हूं, तो para position से they will attack here 2 carbons, 2 molecules of your alpha naphthol. So what you will get in the end? So you will get, so what I will do, you will get, here I will write. So you will end up with this type of structure. And then here you will end up with another alpha naphthol molecule. So you have OH here and you have OH here. तो ये जो आपका मॉलिक्यूल बन जाता है ये वाला, इस कंपाउंड का कलर होता है पर्पल. बिकॉज ऑफ दिस कंपाउंड, अगेन आई विल फोकस हियर, इस मॉलिक्यूल का कलर होता है पर्पल. आई विल नॉट गो इनटू द डिस्कशन ऑफ इट व्हाई एक्चुअली इट इज कलर्ड, बिकॉज इट बिकम्स हाईली कॉन्जुगेटेड. सो ये कॉम्प्लेक्स यहाँ पे बन जाता है व्हेन द furfural molecule this which is formed after dehydration from the glucose, so it reacts to 2 molecules of alpha naphthol, it forms this complex and it has a purple color. तो दो वो जो जंक्शन है उस पे ये मॉलिक्यूल जो है they give this purple color. So the presence of this purple color which comes out from the test tube, that indicates your sample contains biological polymer. So this is our reactions theory and what are the basics involved. So now in the lab we'll prepare, in the next lecture we'll show that video where we'll prepare this and we'll see the ring is formed at the junction of these 2 layers. That indicates the formation of this complex. So हम देखेंगे separate lecture में. So this is all in the theory and description.